From Microbe TV, this is Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from Chevy Chase, Maryland, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. Hello, Vincent. How are you today?
I'm very well. I had a busy day. I was in Princeton planning for the next edition of Principles of Virology. I came back here. I recorded Daniel's TWIV update. Then I recorded Puscast for them, and now here we are. Daniel's in Spain, and his internet is the pits. The pits. You know John McEnroe, he once called the English reporters the pits of the world. Remember?
Yes.
Well, this internet was the pits of the world on the island off Spain. And I'm not sure the recording worked on his end. Well, I can't release a podcast with just me listening, right?
It would be a large amount of dead air, right? Black space.
Yeah, I'll fix it. Don't worry. I'm a good fixer. By the way, let's welcome our moderators for tonight. Let's see. I saw Steph. I saw Frank. I saw Vanity Nutrition. Who else is here? Did I see Les? Tom
Where's
is
Les
here.
and Tom?
Do you see Les?
I don't see nobody.
You know, I don't know if that's not proper English, young lady. Can you rephrase that?
I know that it's not proper English. I don't see anyone.
I don't see Les, but you know, Les, whenever I hear his name, I always think of Les Brown and his Band of Renown. I don't know if you would know that. Wasn't he on Johnny Carson? Wasn't he on Johnny Carson? Oh, there's Les. Hi, Les. Welcome.
I don't know, but I think of Les Newman, the newscaster from WKRP.
Oh, okay.
Because my friend from Montreal, he's fascinated by the flying turkey episode over Thanksgiving. And every year he tells me about it.
So Les said, I saw Les. You saw Les Brown, really? That's pretty funny. Anyway, thanks, moderators, and welcome everyone to another exciting episode of Q&A with A&V, where we have built a little virology community here on Wednesday evenings.
I don't know if I would say it's exciting, but okay.
I think it's damned exciting that we have almost 500 people who are interested in talking about viruses, real time.
That's good.
And so forth. Anyway, did you have a good week so far? Are you getting used to Maryland?
The GPS lady stopped yelling at me on my way to work, so that's good.
Oh, you mean because you know how to get there now. You don't get lost, right?
Right. Nor do I go down the street the wrong way. Nor do I go out the parking garage through the wrong exit.
Yeah, you're learning curve.
And
nor
do the security guards come chasing after me with their guns pointed, going, ma'am, you have to badge out. And I'm like, what? What does that mean? What am I supposed to do?
You know, you were a little too casual at Columbia. So at Columbia, we had these badge swipe thingies, right?
It was totally useless.
And everyone would swipe their badges except Amy. She would walk in and nobody said anything to her. I don't get it. If I walk in without swiping my badge, they start yelling at me. I've been there for 40 years, you know, but Amy gets away with it. Anyway,
they come yelling at you, going, ma'am, you have to swipe badge out. And I had no idea what that meant.
Badge out. It's a new word. Badge out.
Yeah, exactly. So
Listen,
it means that
speaking
if
of
you
my
leave
lab,
after 6.
Yeah, you have to badge out.
Yeah.
Okay.
And nobody tells you that.
So I was up at the lab yesterday. So as everyone here knows, my lab is ended the day Amy left.
Ended? It didn't end. It just moved. Moved to a better geographical location.
But let me just spend a few minutes just speaking about my experience. So, you know, I set up my lab in 1982, and now it's closing. Last week and this week.
It's not closing. It's moved.
I'm sorry, wrong language. Yes, you're right. Last week and this week we packed up like 50 boxes of stuff, my reagents, all kinds of stuff, and shipped them to Amy at the FDA. And yesterday I was there on my own finishing up. I did another 14 boxes. And then I tried to clean up a little because on Monday, a hazardous disposal crew is going to show up and throw everything away in the laboratory. So I wanted to be the one to throw out my lab notebooks. Some of my former people, their lab notebooks are still there. I felt this is my doing. I should throw them out. And I got through half of them, and the recycling bin was full, and I just couldn't do any more. You have to separate the X-ray film from the paper, and some students put everything in plastic inserts.
I know who that was. I know who that was.
And you have to take them out to recycle the paper. What a pita! What a pita that was.
She was a pita.
Oh,
she
the
was
person,
a pita.
the person
she was
too.
A graduate student.
And then they're all filthy and dusty. So I got through some of it, and then I said, "That's it." I left. I said, "Let me go to my sanctuary," which of course is the incubator. And I went downtown, and that's it. I'm done. Let them clean up the rest. The sad thing is, so that's it for my lab. 40 years, and boom, it goes to the FDA. I know.
Your lab just moved.
I know, but I'm not there anymore. I'm not there. You have to understand,
it's
a little bit.
You weren't there for a while anyway.
It's a little melancholic. And then there's equipment around. There's a lot of equipment. There's a $25,000 PCR machine, which is going to get
$25,000.
Yeah, it was, wasn't it? I mean, it was 20 years ago.
It.
I got a lot of use out of it, okay? And there's centrifuges, there's all kinds of stuff. Poof, just gone. And now, a lot of it's moved to FDA, but a lot of it's going to get pitched out. I kind of feel bad about that. There should be some...
Well, I was happy to take some of it, but I was told no.
Well, I sent you some things. I hope tomorrow they arrive and you can open them and have surprises, okay?
I'm super excited.
Anyway, that's
one
thing.
I even got my badge to work to the freezer farm today. And I went to visit my reagents that I took from you and your viruses that I took from you.
Oh, good.
And everybody else's stuff today. I went and then I told somebody that we were going to the liquid nitrogen to make a catalog, and her eyes just bulged out and was like, "We're making a catalog?" I was like, "Yeah, we're making a catalog."
It's so exciting to make a catalog. I'm glad that this is living on in your laboratory. It's great, and I'll get over it. But I just wanted to reminisce for a few moments. When I write my book, my autobiography, this will appear in it. I'm going to write one day a book about my life among viruses. Maybe I should call it that, A Life Among Viruses. How's that?
Well, yeah. Why wouldn't you call it that?
And I hope everyone here buys it, okay?
So how much of it is going to be about the 20 years of you and me?
Nothing. No, nothing. You're not even going to be in the book. No.
Okay. Okay.
Maybe I should have a 2-volume set and do the first volume up to Amy. It gets published. It's a bestseller. Then I die, and the second part never gets published. How's that?
Okay, that's fine.
Is that a plan?
That's fine.
Anyway, I am going to write a book. I've been talking about it for years. It's going to be like one chapter on some virus history, one chapter on Vinny history, one chapter on virus history, one chapter on Vinny history. You got the pattern there, folks? Okay, so that's
fine.
The other
so
thing.
Then at the end, it'll be a chapter on Vinny history, Amy history, and virus.
No, there's no Amy in this book. I already told you.
There's always Amy in the book.
Okay.
You don't seem to okay, fine.
You can edit it.
No, I don't want to be in the book. Forget. All right,
what, now we're turning around and being petulant. We're going to be petulant, is that right?
No, I'm going to delete myself from the book, but I will edit it.
I wanted to
make sure
I have a sticky here with some things I'm supposed to say tonight. Can you imagine I made a sticky?
Holy shit.
I got through one thing.
You made a sticky?
Yeah, I wanted to not forget these things. All right. Okay. I made a mistake last week, okay? I want to correct it. It's not good to make mistakes.
Okay.
Gyneos, G-Y-N-N-E-O-S. This is the Bavarian Nordic. It's a smallpox vaccine. I said it was a monkeypox vaccine, but it's actually small, and it's an attenuated vaccinia virus, or not really, but it's not a monkeypox vaccine. It's a vaccine for smallpox that will work for monkeypox. Okay, so Gyneos, I said it was a monkeypox specifically. I was wrong. Okay, so that's corrected. The other thing I wanted to tell you is that So I went to Princeton to start working on the 6th edition of Principles of Virology. Some of you have that. Oh, this is cool. Amy, I didn't tell you this. Listen to this. Anne and Jane gave me their extra copies of POV.
Ooh.
And everybody signed them. So I now have 10 copies of POV signed by all 5 authors. And later this year, we're going to do a fundraiser with those.
That's cool. That's almost like the Rod book. Do you know what I'm talking about?
Yeah, the one with all the famous people in it.
Yeah.
Anyway, the new edition, the 6th edition, is going to have a new chapter. And at the end of the book, we're going to have 4 chapters all related to pandemics. We're going to have evolution, emergence, HIV/AIDS, and COVID to try and make a cohesive thing.
Why isn't there an HCV chapter? It's a pandemic.
I brought that up, HCV and Hep B, and I said, what about them? And they said, we're going to have to address that in the chapters, and we're going to have to define what a pandemic is. And we all decided we're going to make up what a pandemic is. The hell with everyone else. The hell with the WHO. We're going to define pandemic.
That's fine, but I'm still upset about the hepatitis pandemic, which is not included.
No, we're going to include it. It may not be a specific.
No, you said it needs to be a separate chapter. Why is it not a separate chapter?
It's not going to be a separate chapter.
Why?
Not enough people. How many people?
What are you
70
talking
million?
about?
70 million? 70 million people, right?
No, it's way more than that. Well, way more than 70 million people. I can ask the woman. But it's way more than 70 million. She'd be insulted.
Well, anyway, look, here's the reason. HIV is the most studied virus, so that deserves, and a huge pandemic, so that deserves a chapter. COVID has just screwed up the world, so that needs to have a chapter. And then that's already too much space. We don't have enough space, so that's why we put other. But you can't do everyone. It's not supposed to be comprehensive. It's just supposed to do some illustration.
Well, there's no principle. There's no principle after HIV then.
Yeah, there are going to be some, but I agree there are no principles. However, it has revised a lot of our thinking, and we can point out that there's so much left to learn that there aren't new principles yet. And then at which point we all said, what other principles are there? Don't we know them all? I don't know. But anyway, I thought you would like to know this. Wednesday evening, listeners, you heard it first here. There will be a COVID chapter in POV6, and that means
it's 250 million people who have chronic hepatitis B. A quarter of a billion people.
I'll make a pitch. I'll make some kind of a pitch. But that means I will have a COVID lecture in future virology courses. Okay?
Mm-hmm. And the only reason why COVID is such a big deal is because we were completely ill prepared and we polarized it.
Of course.
So that is not a scientific property. That is a political property. And it is not my fault that people don't understand that just because you cannot assess antigenic drift by your specific assay doesn't mean that it doesn't occur.
Of course.
Well, no, it's not, of course, because you claim that one of the greatest things about this pandemic was learning that SARS-CoV-2, that coronaviruses undergo antigenic drift. And I'm saying,
Amy, if you haven't looked for something and you don't know it, when you do discover it, it can be a principle. It doesn't matter that you've ignored it.
It's not a principle. But it's not a principle. Antigenic drift is antigenic drift. It doesn't matter how it occurs. If it occurs on measles,
what you're saying,
or for polio, or on
what you're saying
coronavirus.
Is not a big deal because it happens with common cold coronas. But we didn't know that.
I'm saying it happens with everything. I'm saying it happens with everything. That for being a principle, it means that it's applicable to almost every entity.
Okay.
And so you can't say anything about.
Look, we can't have a chapter on every pandemic because there are so many, right? We got HIV.
You're not even counting the 1918 pandemic.
Well,
you're totally
COVID,
skipping
we have
that.
Hep B, we have Hep C. We can't have a pandemic.
Right, but
you write a book on pandemics.
Right, but you're even skipping the 1918 flu pandemic.
Oh, yes, I agree. I think we have to bring up aspects of these in the chapters, but we can't do them
Well, I
all.
don't think you should have a chapter, therefore, solely on 2 pandemics.
Well, that's what we decided to do, and you weren't
Not
there,
good
so I'm
decision.
sorry. Okay?
Not good decision.
All right. Two
It's
more
fine.
things, folks, before we before we get to your questions.
Yeah,
All
this
right, Paul.
should be called The World According to Amy, this podcast, because The World According to Amy works well and makes sense.
Okay, you want to change Q&A with A&V to the world according to Amy? It's fine.
No,
Okay.
not really.
Two more things, and then we'll get to your questions. Isn't Amy always right? I think she's right 99% of the time, and I'm looking for those 1%. You should have been a lawyer. Yeah, someone just said you should have been a lawyer. Why don't you go to law school?
Why do I want to be a paper pusher?
Okay, two more things. First of all, we have Paul Offit on Friday on Twiv. What do you want me to ask him, folks? Put your questions in the comments here, okay? Vincent, you know Amy's always right. No regrets. Listen to her. She's a much younger generation. Okay, she's always right. But are you telling me, and I wish I could curse here liberally because I feel like cursing. Are you telling me that 40 frickin' years of experience in virology doesn't mean anything? I don't know squat. Are you telling me that? Come on, I know something. And that's what when I said to her, and when she used to work in the lab, I said, why don't you have some respect for 40 years of research? No, you're wrong. You're wrong. Is this a death of expertise, Dr. Rosenfeld?
No, it's not a death of expertise. It's not a personal thing. You're taking it as a personal thing. It's not a personal thing.
I never take anything personally, Dr. Rosenfeld. Nothing.
Yes, you are. You're taking it as a personal thing.
No. Now, Miguel is offended that you call lawyers paper pushers. You better be careful who you offend. And put your questions for Paul Offit in. So Jeff
they don't do any experiments.
Jeff has put the first one in. So put your questions in, and we'll get to them. And then one more thing. Okay, this is really important. Okay, folks, I'm going to share my screen for this. All right, let me close this thing. This is important because, I'm supposed to teach a course in the fall called Viruses Live, right? All right, what's going on here? All right. Viruses Live.
See.
You can't see anything? Oh, I have
it's
the nice...
Small. It's
too
small.
It's too small. All right, let me try and... put your glasses on. Okay, Viruses Live. This is my suggestions for...
Okay, so you got Picornas, Coronas, Calices, Flavies, Togas, Hantas, Bornas, Influenza, Philos, Paramyxos, Raptors, Arenas, Bunyas, Rotas, HIV. Why is HIV and HTLV separate? They're both retroviruses. They should be the same.
Okay, fine. Now, doesn't matter. DNA viruses.
Herpes, adeno, pox, papilloma, polyoma, parvo, circio. Well,
so these are...
Where are the alpha viruses?
Alpha?
Yeah, alpha.
Aren't they togas?
Aren't Flavies togas?
No,
Flavies are togas.
No, they're not togas.
Yeah, they are.
They're Flavies. It's a Togaviridae and Flaviviridae are two different families. Okay, that's one time when Amy was wrong, okay?
Okay, fine.
So this is a human-centric course because if I put in viruses of bacteria, plants, and fungi, it will balloon into an unmanageable entity. You know what I mean? What do you think of that? Do you agree, Amy?
Yes. I just don't agree that HIV and HTLV are different because the truth of the matter is the picornas, you got the entros and then you got the alphas and the rhinos, and that's a whole different issue, right?
Yeah. But is there anything missing? Is there anything you'd like to see here? Rabies virus. Yeah, rhabdoviruses are here.
Rhabdos are rabies.
That's rabies.
Wait, did you have, I guess you're considering measles a paramyxo?
Yeah, I'm not considering. It is.
And, well, what about like rubella, or morbilloviruses, or morbilloviruses.
Rubella is a yeah, it's a toga virus actually. It's a different family from the paramyxos. Anyway, so think about it, folks. Get back to me because I have to develop this course, and there's a lot of viruses. I mean, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 18, 19, 20, 22 different virus families. I mean,
So you're going to go on for 44 weeks?
it's too long. It's too long. So I'm not
Well,
sure what to do about that, you know? I mean, some things are going to have to be brief, but those should it be a human virus course? That's the question. If not, then I have to not do everything. I can't do every virus, obviously.
well, not only that, but like rhabdos, the really big one is rabies, right? Whereas
Yeah,
for
yeah,
herpes,
sure.
you're going to talk about herpes simplex 1, 2, and chickenpox,
Yeah.
zoster, right? And ad is really just ad. It's one big ad, right? There's different serotypes, but it's really one big ad.
Yes. One can focus on one ad. Yes, that's absolutely right.
Right. But like, what would you do for picornis? Because then you have the rhinos and the enteros, and you have the echoes and the coxsackies,
Yeah, I mean, I
and then
think,
you'd have so you don't want to just solely focus on polio.
well, I think part of each lecture is a little history of when the viruses were first identified and then leading to the modern day situation. So for picornas, it's polio, right? The first one. But
Right, but
I
then very rapidly after that was coxsackie and stuff, and
so
then
so
in the
yeah, and so you can imagine. Okay, the current day situation is we're trying to eradicate polio, and now all these other viruses are clearly very prevalent. And here's what they can do. What's the pathogenesis? What can we do about them? I think for picornas, it's quite straightforward. But do you think
I
that
don't know
do you think a good way to do
where's
this?
rhino fit into that.
I think it's a respiratory picornavirus, right? I would keep it separate when you talk about pathogenesis. But is a good way to do this is to say I have 30 lectures and I have to fit this topic into those 30 lectures. That's the question.
I don't think so.
Zika virus would be a flavivirus, so that's going to be covered. And I don't want to make it so diluted, so expansive that we don't spend more than 5 minutes on each virus. That's not useful, right?
Right, but but if you do the picornas, where are you going to put Hep A?
It's a picornavir day, right?
Right, but you start off with polio, and then you want to go. Then you could argue about Coxsackie and echoes because Sabin identified the majority of echoes, right?
Mm-hmm.
So and they're gut viruses, and they kind of are the same. And then you could do all the rhinos from the 50s and blah blah blah, right? So that's like 4 lectures right there. But
It's
then
got
the
to be
fifth
two. No,
one,
no, no, no. It's got to be 2 lectures. It can't be 4.
okay. Well, then the third one is addressing the Hep A problem because
Why
Hep
do I
A
need
is
a?
totally different.
I don't need a whole lecture for Hep A. No, the picornas have to be 2 lectures.
It's got a vaccine.
Yeah, I think we have to do 2 lectures per virus family. Otherwise, even at that, that's 44 lectures. That's a long course, you know. And anyway,
Yes.
do a virus
44
bingo.
lectures would be 14 weeks. If you do 3 lectures a week, it's 14 weeks.
I'm not sure people would come to 3 lectures, although it's recorded, so they don't have to come to all of them. It's true. Anyway, someone asked, do you have to take virology live? Yes, you should take virology live first. Many of you sat through it, but you should do that. Otherwise, you're not going to understand a lot of this. All right, we'll revisit this, I'm sure. But I seeded the what?
Have
What did
you
I?
have you made an outline?
That's my outline right there.
That's just a list of viruses.
It's the beginning
I
of an
want an
outline.
outline. I
Hey,
want the
Amy, I'll
outline
get
of.
it. I will do it. Don't
Okay.
worry. All right, here's some questions. Now let's get to the folks. They're a little antsy because they're being ignored. Philip says, this is for you. I understand smallpox.
This is not an ignoring of them. This is just a dialogue.
I'm just kidding. Smallpox is more deadly when it enters the lungs than the skin. Does this apply to monkeypox?
No.
Monkeypox is generally transmitted by contact.
Right.
And you will get it if you're right in someone's face, I believe.
Yeah, but nobody's really died so far from monkeypox.
No, not this round. We're up to over 6,000 cases now, right?
I don't know. I haven't been keeping track.
Yeah,
but I do know that New York City is either out of vaccine or afraid that they're running out of vaccine, so they tried to order more.
Monkeypox? You mean the smallpox vaccines?
Yeah. I never thought that there was a monkeypox vaccine. I don't know why. Never thought that. I thought it was always a smallpox vaccine.
It was always. There's no monkeypox vaccine,
right,
as far as I know. Rob, thank you, Rob, for your contribution. $13.99. Interesting number. What's the significance? James Webb Space Telescope is fully operational, but will it detect signs of exofungus in the atmospheres of distant worlds? The light will reach your eyes on July 12th. That's very exciting.
It is very exciting. You can ask Dixon and Condit because they're all into the telescope.
Yeah. Are you into it, Amy? Do you like this space stuff?
It's fine.
It's fine.
That's fine.
Was COVID-19 vaccine the most rapid vaccine development in human history?
It depends on how you define development.
Look at the polio vaccines. Even if you start with Salk in the 40s, it took 10 years to develop it. Same for Sabin.
Well, I mean, the fact is that I'm not clear that we Kaprowski has attenuated vaccine way before Sabin. He was drinking it as a cocktail.
I just go back to the original inactivated flu vaccine developed in the 30s or 40s by, among other people, Salk. And that was developed for the Army first, inactivated vaccine. I'm not sure that took so long. I don't think it took 10 years. But a year... I mean, it's a little bit misleading, a year for COVID vaccine.
That's what I'm saying. It depends on what how you consider it.
So, I mean, all the preliminary work, showing that you could make RNA that wouldn't be too antigenic, too sensed by the innate system to be a problem and so forth, that took a long time. And then they tested other vaccines right before COVID, correct, Amy?
Yeah,
but in animals.
But it's not just about the RNA being antigenic. It's the lipid nanoparticle by the guy in Canada. It's about the T7 by Pagano, blah, blah, blah. I mean, the truth of the matter is it's really taken 50 years.
So if you define development as day one, they say, let's make a COVID vaccine to deployment in people, then probably this is the most rapid one, yeah. But if you include all the pre-development that you need, then it's not. What does everyone think about BA5? Worse or same amount of disease as previous BAs? Dr. Rosenfeld?
The same.
The same. Remember when Omicron first arose, everyone said it was less severe?
Yeah, because they're idiots.
Thank you, Michael, for your contribution. There are some papers that conclude that BA5 can cause infection in the pulmonary alveolar cells. Others say the opposite, that variants 4 or 5 remain confined to the upper tract. And this is the problem with doing these experiments in cells in culture. I assume that's where these have been done, right, Amy? Or in mice or golden hamsters. right?
No, I assume that they're actually done in culture. In mice and golden hamsters, it's pretty clear. You can pretty much tell where it is.
Yeah, but then in golden hamsters, Omicron originally was said to be attenuated, right? And that's clearly not the case in humans. So,
Doesn't mean that the tissue tropism was different. Just means that the hamster might have had a more robust immune response. Maybe it's
yeah,
like the bat. It has more than one type of interferon. I don't know.
I think you have to be very careful. I mean, human experiments are difficult because you're not going to get much information on the lower tract. And then animal experiments are problematic because you don't know what they mean for humans. So I would default to they're pretty much all the same. I would say in the end, maybe in 50 years, we're going to conclude that all the SARS-CoV-2 variants are of equal virulence, and it's just the immune response that modulates it. Is that a fair statement, Dr. Rosenfeld?
Yes, but I already made that argument when I corrected the people who said that symptoms occurred faster, right? I said, no.
Let's do a few Amy comments. I hope you're
We
getting
don't need to
adjusted
do
to your
Amy
new
comment.
location. Okay, why don't you just see people are thinking about you. I'm thrilled you like the new position. I'm sorry you had to move away. Your family and you must miss each other.
Sure, we could go with that.
Amy, I just went to the Bata Shoe Museum. I live across the street from it. Couldn't help to think of you when I saw the Manolo. What's the Bata Shoe Museum? Do you know, Amy?
No, but now I'm in. I want to go.
Where
Where
is
is
it? You
it?
don't even know. Bata Shoe Museum. And then, it's in Toronto. You're not going there. You got to stay there and work. You're not going to Toronto.
Oh, okay.
You said our immunity was vital given reproduction is successfully met. Yes, so let me clarify that first. So I believe that we and other organisms evolved so that our immune system would protect us through reproductive age, and then after that, nothing matters so because you're not reproducing anymore. Okay? People are living a lot longer. Has evolution not kept up with the science? Yes, because the increase in length of life has only occurred from like 1900 to the present. Not enough for large-scale evolutionary changes in our biology. So yes, it's not keeping up, but maybe it takes thousands of years for these changes to propagate through humans and to be in all humanity. This takes a long time. So no, reproduction has not kept up. That's why we get cancers. Our immune system senesces as we age and so forth. Look at Mamie's lovely smile. See, they like your smile. Give us a smile.
Good.
There it is. There it is. Right. What's different about
Yeah.
this monkeypox outbreak that has it spreading so widely and quickly? Well, that is the question, isn't it? And if anyone tells you they have an answer, don't believe them. Do you have an answer, Amy?
No.
No. Amy would not, say something that's not correct. Just about the most honest person I know. There's no.
There's no need to lie. What did Mark Twain say? If you lie, you always have. If you tell the truth, there's nothing to remember.
I told you that first.
Yes, you did.
Yes. I'm glad you remembered that. It's a good thing. If you tell the truth, you don't have to remember anything. I don't know what's different about this outbreak. Don't tell me it mutated, right, Amy? We don't have any information on that.
I have a feeling it has more to do with like a naive population.
I think it got into a naive population, and it's being transmitted in that population. But why that's the case, I just don't know.
I'm not clear how it entered. I think it will be interesting to understand how it entered.
Yes, I'm not sure we'll ever understand that, but okay, now BA4, BA5, can we be comfortable with our degree of protection? So this is difficult because actually Daniel Griffin in tonight's TWIV update, he just discussed briefly a Lancet paper showing protection of 3 doses of vaccine of Moderna, Pfizer, I think it was Moderna, against Omicron, against hospitalizations, 86% vaccine efficacy against hospitalization. So it's still good, yes. But 86 is not 100, so that's 14% is a lot of people when you're talking about millions of people getting infected. So some people are going to get hospitalized, and some may even die.
Wait, how are they counting the hospitalizations? Are they hospitalized due to it, or they came to the hospital with it, or is it all in one?
That's a good question. I'm not sure they
So
distinguished.
So
they didn't
before you
distinguish.
start saying, so before you start saying that 14% is a big number, you have to define what you're saying.
I mean, you're saying it could even be in the
fair.
90s. You're saying it could even be higher, right? Yeah, I agree. I agree with you.
It's not a fair conclusion.
Now, and Daniel made the point, which is a really good point. He said, a year ago, we were being very conservative at admitting people to the hospital because the hospitals were overburdened. And now he said there's plenty of hospital beds. So if you have an elderly person with comorbidities, you may admit them with just the slightest symptoms because you want to be careful. And that's not really reflective of the severity of the illness. You know what I mean, Amy?
Yeah, I do. So I don't understand what this statistic means.
So anyway, all that is to say, Tom, I feel very protected with my 3 doses of vaccine, which is not to say I may not be infected at one point and I may not feel very good for a couple of days, but I'm confident that once my memory cells are activated, I'm going to be good. And I think Amy is confident too, but I know that's anecdotal, but I'm just telling you how I view the data. Amy, what would happen if you took your thymus out of the body?
It involutes to begin with, so it doesn't do anything. So
Adults, once you're born, you don't need it anymore, right?
Apparently not.
Well, there you go. It's a small organ, so.
People used to take out your tonsils, and it was like no tomorrow. So have you died yet? Because you got no tonsils.
I have my tonsils. I never had them taken out. Did you?
Oh. No, but like my mom did.
Is having big tonsils a good thing? It depends if they're good. If they're big, like for a few days at a time, that means you have an upper tract infection. I can tell when I have a rhinovirus or some cold, my tonsils under here get swollen. Excuse me.
Those are lymph nodes. Those are not tonsils.
I'm sorry, lymph nodes, yes. I know, those are the lymph nodes. The tonsils are in there, but they're the same. They're the lymphoid organs that are draining the upper respiratory tract. Yeah, so if they're big, is it a good thing? I don't think it's necessarily good. It just reflects human variation. I just tickled my throat.
Physiology is not your strong point, is it?
I don't have any strong points. I'm a good teacher.
That's not true.
I'm a good teacher.
That's a strong point.
Do you ever do a TWIB? What's TWIB?
What
This
is a
week
TWIB?
in what? Bacteriology?
That's TWIM.
That's part of This Week in Microbiology, right? Bacteria are microbes.
Everyone on TWIM is a bacteriologist, but you.
Oh, poor Eddie. Hopefully this live stream will cheer me up. We found maggots in our outdoor trash bin, and then some moron rear-ended me. I'm so sorry. That sucks when you get rear-ended.
Yeah, it does.
That's why I'm trying to drive less. The maggots.
I understand.
That happens.
So Jamie Morrow is tired of me.
Don't amplify such comments, Amy. We'll just have them deleted, okay? We're not tired of you. Don't worry.
It's okay.
Jeff wants to know, is there a point where we won't need to be concerned with spillback from animals? I don't know that you can ever not be concerned, right? You'd have to look at every animal to know the threat, and we just aren't doing that. So at this point, we don't know who is infected out there and to what extent, right? We know a little bit about the deer. What do we know about wild rodents? Very little. I think Tony Schounts is working on it, and I'm going to go to a bat meeting in July that's this month. in Fort Collins. Anybody from Fort Collins? I'm going to be in Fort Collins for a couple of days. And,
you and Brianne.
What was I going to look up here? I was just going to look up something related to this question. Oh, well. I don't think, Jeff, you're going to stop worrying for a long time. Pete's on mobile data. You know, last week, Amy did this stream on her phone by tethering to her phone. How about that? And it looked pretty damn good. Tonight, you're back on internet because I understand that your internet tech appointment figured out the problem.
Yes, my internet tech appointment went very well. He was most helpful. And there was no yelling or screaming or anything. We did something. We pushed some buttons. We deleted some numbers. We added some new numbers. And wham, bam, thank you, ma'am. Voila, there was internet. Much better.
Will the WHO declare monkeypox a pandemic on July 18th? No. No, it is not. I think one of the key aspects.
Well, since you guys are going to redefine pandemic, what are you defining it as?
Okay, so here's a phrase that we came up with, and you're going to hear it first tonight, okay, on this live stream. A pandemic affects everyone, but does not infect everyone.
What?
It has a global effect, right? Many people's lives are disrupted, but not everyone is infected. I really like that.
How has your life been affected by HIV?
HIV?
Yeah, how has your life been affected by HIV? You said it affects everyone, but not everyone is infected. So I want to know, how is your life affected by the HIV pandemic?
Well, I didn't get grants because a lot of money went to HIV research.
That's ridiculous.
No, well, listen, you asked me. That's my answer. Don't say it's ridiculous, okay? It happens to be my honest answer.
How is your life affected by the HCV pandemic?
It doesn't have to affect everyone, just a lot of people.
Yes, that's what you said in your definition. It affects everyone, but does not need to infect everyone.
Oh, it affected me because I devoted a good amount of my teaching time to hepatitis C virus. I mean, it's not a negative way. It doesn't always have to be a negative way.
But that's not... So everything's a pandemic then. Everything's a pandemic. I'm affected by people who have rhinovirus, whose kids have rhinovirus, because then they have asthma exacerbations and it interrupts my workflow. So I didn't get infected, but I got affected by rhinovirus. So the rhinovirus, therefore, is a pandemic.
I made a huge mistake in bringing it up. I thought it would be productive. But Dr. Rosenfeld is attacking it, and it's not useful.
I'm not attacking it. It is useful. I just want to know. It is useful. I just want to know how then
all right, maybe it's not a great
pandemic.
Maybe it's not a great definition.
Maybe you need to refine it.
Look, you get the point that it can affect far more people than it infects. Maybe that's a better way of looking at it.
Well, yes, but that is not what you said. You said it affects everyone. So, therefore, rhinovirus is the world's greatest pandemic ever.
It doesn't kill anyone.
Doesn't need to kill anyone. Infection does not need to kill people. You just said that they need to be infected. But
so
it
you're
affects
saying
everyone.
That a pandemic doesn't have to kill anyone.
Right.
Oh, come on. That's the cardinal point, is that you're afraid that the virus is going to kill people.
It's not a cardinal point.
So you would have
a cardinal point.
You would have a pandemic of just lots of but rhinovirus then would be a pandemic, but it's not, Amy.
Well, it is a pandemic.
No.
Yes.
No.
It is a pandemic. Remember the lady who
Excuse me, Ann Palmenberg gave a talk at ASV entitled "Why Rhinoviruses Do Not Cause Pandemics."
Not correct. It's not.
Well, you better call Dr. Palmenberg and tell her it's not correct. Are you going to do that?
I don't need to call Ann.
No, because you don't want to deep six your career at this point, right?
It has nothing to do with whether or not I deep six my career. I'm not calling Ann because of other reasons. But the end is you and I both agreed that she was wrong.
I still am not. I better run the pandemic definition by you when we do it, okay?
Yeah, because this is not going well.
All right, Dave wants to know, are there studies that show that vaccinated people are less likely to spread?
Yes, I just sent you that paper
All right, so spread is
this
hard,
morning.
but a surrogate for spread is shedding, okay? If you measure shedding of an infectious virus, but very few people do that.
I just sent you a paper this morning.
It's probably the one that Daniel discussed today.
Yeah, because I sent it to both of you. What do you think? I'm a lunatic? Of course I know what to do. Hey, I'm exhausted.
Where is your thing from this morning? Did you send it to me or to me? And
I sent it both to both of you. It's called The Effects of Vaccination on Household Transmission of SARS-CoV-2 Delta Variant of Concern, Nation Communications. And if you go and read the abstract, it tells you at the end. It says, our results demonstrate that vaccination reduces susceptibility to infection as well as infectiousness.
All right, here, let's take a look at this. Let's bring it up so everyone can see it. Let's do our screen share. So I just got yelled at for not answering questions quickly enough, Amy. They said, you need to answer some questions. So I'm not going to talk to people anymore after this. I'm just going to answer questions, okay? So here it is. Effect of vaccination on household transmission of SARS-CoV-2 Delta variant of concern. Let's look at the conclusion. Vaccinations reduce susceptibility to infection as well as infectiousness, which should be considered by policymakers when seeking to understand the public health impact of vaccination. So unvaccinated secondary cases with an infection and exhibited a threefold higher viral load, and that's RNA, so I don't really buy this kind of study because viral load by PCR is not really telling you anything. My feeling is that you can find studies that say there's no effect of vaccination on transmission, and that would be, I wouldn't buy that. And then you can find the Geneva study which says there was an effect of vaccination on transmission. So, I mean, how could there not be any effect? Isn't that absurd? There has to be some effect because you're reducing shedding and not only amount but time as well. Okay, where was I? What was the last comment?
I like the fact that people have decided that I'm in a mood and blah, blah, blah.
No, Amy's not in the mood. Amy's fine tonight. What, did you know I was self-absorbed because I didn't ask you how you were? Oh, here it is.
It's because people are crazy.
Okay. I'm having COVID rebound. Have you heard if round 2 is usually milder than round 1? I haven't heard anything, but here's what I found out today. That most rebounds are not hospitalized. So if that's any consolation, that would be fine. Amy looks positively happy. See, there are people who say you're in a mood, and there are others who say you're happy.
Yeah, so there's also people who say I'm condescending.
No, you're not condescending at all. There's a small group of double envelope plant viruses. They replicate in factories in the cytoplasm. I'm confused about where they pick up a double envelope. Well, they could bud out of the factory and pick up one, right? And then they could...
Well, couldn't they bud out of the nucleus and then bud through the Golgi? And
but
then...
he says they replicate in the cytoplasm, though, so they're not in the nucleus.
Oh.
They could bud out of the factory and then bud at the plasma membrane, right?
Yeah. I mean, I don't know where the factory is. Is it like the factory? Is it like the lipid droplets of picornas, or they are translated and assembled in like the ER to the Golgi like a corona? That would be double membrane.
It could be. Yeah, you could go through ER. That's right. Les Brown was Bob Hope's band. Oh, okay. I didn't know that. Les Brown is Band of Renown, which just came into my mind.
Were you a Bob Hope fan?
No, I wasn't a Bob Hope fan. No. I mean, you liked the Rat Pack, didn't you?
Yeah, but I didn't think Bob Hope was the Rat Pack.
No, he wasn't.
That was Frank and Dino and who was
Sammy
the third
Davis.
one? Sammy Davis. Yeah.
Yeah. No, I wasn't a Bob Hope fan. What was
And
the
Peter
hardest? Peter what?
Peter Lawford, the one who was married to a Kennedy.
Okay. What was the hardest thing to throw out from the lab, Amy? I don't think you had any hard thing because you were moving to a new thing, so it didn't matter, right? For me, the hardest thing to throw out was Amy.
I got thrown out into the garbage can. You packed me up. Did you put me in the recycling or the autoclave?
Let's see. What was the hardest thing to throw out? So I threw out a lot of viruses. I threw out a lot of cells. That didn't really bother me because a subset of everything was going with Amy to the FDA. I think, it's funny. This morning I woke up and I said, shit, I wish I had brought those little racks, an example of those little racks. So I saved a gel box because I want to put it behind me in the incubator. It's a protein gel box. I want to put it behind me as an example of equipment. I had these little metal racks that I got at MIT where you could take a part of it and clamp it to it so you could boil it.
What are you talking about? I have them. You want them? I can give
We left some behind. Did you take a bunch?
I'll give you. Yeah, I have. Actually, we didn't leave any of those behind because you didn't have very many, and I've had them for a while. But
there were a few left behind, and I was considering going up tomorrow morning and getting one, but I don't think it's worth the trip. But that's trivia.
I have them.
Actually, the hardest thing for me to throw away were the lab notebooks, because that's people's data, and I think it's sad to throw it away, but there's no use for it anymore. If it's not published, then that's it. What happened to the mice in the mouse house? Well, 3 of them went to the FDA, right? Or 2. How many did you take with you?
10.
Yeah, so 10 got new home, and the rest, sadly, were sacrificed.
Well, a lot of them were sacrificed as part of the experiment. The last parts of the experiment, the terminal bleed, sacked like 2/3 of the colony. And those serum are now sitting in my refrigerator.
Yeah, they were all properly used. We don't waste anything. Do you have any idea how long the lipid sac which transports the mRNA vaccine, I presume, lasts in the body? My understanding is it lasts a few days, based on half-life studies in mice. The nanoparticles, Amy.
Oh, I was like, what is that? Yeah,
the lipid nanoparticles. Will it continue to be causing spike to be produced until it's destroyed? Well, they have to get into cells to produce spike, right? So once they get into cells, that's it. And if others circulate for some time, they will they will be taken up by cells in your body that filter out particles eventually, and they will be destroyed. And so that's where the couple of days comes from. Ooh. I didn't know this. A badger is someone who won't let you alone until you badge out. So you got to remember that, Amy.
Okay, I'll remember.
I've only received my primary series of Pfizer. I've been hesitant to get the booster. Almost everyone I know has been vaccinated and boosted are getting COVID. Yes, but they're getting milder COVID most likely from being boosted. So I think boosting is a good idea. A friend, not vaxxed, second COVID, has scarring on the lungs. Will this be better, less harmful? You mean a second COVID infection? I can't imagine. Well, it depends whether his immunity will prevent the virus from going down into his lung and causing further damage, and no one knows whether that'll happen or not. No one can tell you. Focus on neutralizing antibodies only. Does immune evasion mean the shape of the antibody doesn't match the epitope? Theoretically, should it neutralize BA5 better if we increase the level? No, increasing the level is not going to increase neutralization, right, Amy, if it's resistant.
Right.
No, so it changes the shape. The epitope shape changes, so the antibody doesn't bind effectively any longer. So simply adding more antibodies is not going to do it. However, Amy, let me qualify that answer. The FDA has increased the dose of Evusheld because BA5, BA4-5 are somewhat more resistant. So they doubled the dose to make up for that. So I guess if the evasion change in the spike reduces the binding, maybe by adding more antibody, you can compensate for that. But if it eliminates it entirely, then adding more antibody is not going to make any difference.
Right. That's true.
How much risk does an unvaccinated person pose in terms of exposure to an under 5-month-old once they get the vaccine? Who's getting the vaccine? The unvaccinated person or the under 5-month-old? I guess the under 5-month-old is getting the vaccine. The full course of vaccine, I think minimal risk, but I can't quantify it for you. And I don't think Amy could either. Could you?
No, I won't even attempt. I don't think it's a quantifiable entity.
If it's a ddPCR machine, I will take it off your hands. Unfortunately, you can't have it because it's barcoded. It has the Columbia barcode. Not that.
What is a DD?
Dideoxy?
Why is how do you make a PCR machine a DD? I don't
know
understand.
if DD. Let's make sure. Oh, Biorad DD PCR. Nanoplate digital PCR. Do you know what that is?
No. But
Sure,
I guess
Jeff will
it just
explain
takes in a nano. I guess it just takes a nanoplate instead of regular 0.2 tubes.
And by the way, I'm giving Amy stuff just so it has a home. She could get everything she needs at the FDA, except for viruses. I have some unique viruses. But
we have a lot of unique viruses.
But she could buy everything herself at the FDA. But why should I throw it away? Might as well give it to her, right?
That's what I argued when I got here.
Yeah. Do you guys know how to use a centrifuge? Yeah, of course we do. I mean, we.
Did you know that there's centrifuge training?
Oh my gosh.
Seriously? Today I took centrifuge training. I took autoclave training. I took budget training.
It's amazing, all this training that you never got in my lab, huh?
I took freezer training.
You took badge training, I guess, right?
Yeah, I took a lot of training.
Sorry to hear, Vincent. I hope all is good. You get your memories, and I bet a lot of students enjoy your efforts. Yeah, no, it'll be fine. I'll be fine in a week. Just a tiny bit bittersweet. But I still teach, right? And I get to excite students, not just at Columbia, but online, right? We do our live courses. So no worries. I'm good. News from Australia. Recommending vaccine number 4 due to spread and hospital admission. Winter effect. It's winter down under, yeah. Is virus growth inhibited in heat waves? Well, it's more easily inactivated, right? And for viruses that transmit through the air, if it's warmer, that will impinge upon transmission. But yeah, now is your respiratory virus season, so that's why they're doing that. Any logic? Thank you, Michael, for your contribution. Any logic to roll out a vaccine targeting Omega variants when they have been outcompeted? I think not. I'm going to ask Offit that on Friday, but he's already written it in his opinion piece. Where did that come out? In Stat. He said it's kind of, where the other viruses, BA1 is in the rearview mirror. I think that's how he put it. You like that analogy?
Yeah.
Dr. Rosenfeld?
Yeah, but I think Jerry Weir had a brilliant point when he said, predicting what for flu Because you're doing the same thing, right? It's already in the rear view mirror, right?
Mm-hmm.
And we use some kind of prediction algorithm for flu, but it's been honed for 400,000 years, whereas it's not really been honed.
Everybody likes the title, A Life Among Viruses, so that will be it.
I think it's great.
And Rach is going to buy it. Rach, I know she's from the Isle of Man, where they have bagels. I remember that.
Yeah, it's good.
You're only 69? Hey, I don't look older. Give me a break. I look like 54, okay? Except for the gray beard. I know the beard.
No, the beard doesn't. No, the beard is fine.
Not a single gray hair. I mean, there's not a lot in the back, but... Sorry.
That has nothing to do with it. But the beard is fine. The beard doesn't make you look older. Don't be ridiculous.
But the book will be understandable, so you don't have to worry about that. I hope Vincent doesn't go Walter White with all that glassware. All right, what is Walter White? Do you know? Is it a movie? Walter, what did he do?
You're asking me if it's a movie? Seriously?
Oh, the guy from Breaking Bad. Okay, I guess
what
he
is
smashed.
Breaking Bad?
It's a TV series about the chemistry teacher who made drugs, made a lot of money.
I never saw it.
I think there's a scene where he breaks a lot of glassware.
I don't know. I didn't see it.
Well, I watched 4 episodes and I got bummed out because it's too violent.
I've never saw it.
So it's my memoir. Yeah, exactly right. That's what it is. It's a memoir. It's not an autobiography. I called it that, but that's not correct. Thank you, Jay Villa, for your contribution. I emailed you a few days ago about my daughter and remdesivir. We are both feeling really good with little symptoms. Very good. Thank you for your contribution. I appreciate it. These are the new variants. Did you know BA is before Amy and AA is after Amy? So that's my lab.
It's good.
I believe in the vaccines, but I am disappointed. It seems that even with vaccines and boosters, COVID is causing symptomatic disease. Now, B4 and B5 are escaping neutralizing antibodies. Well, actually, the original Omicron was also escaping neutralizing antibodies. These just more. But, all right, so there are 2 issues here that Amy, I'm sure, would point out. One is that no vaccine prevents infection. But yes, the Omicron variants are causing a little more disease. You're still not dying, okay? They're protecting you against death. And I think severe disease is much less.
Here's my question.
Yep.
So if you're disappointed in this, the person must be on the verge of a nervous breakdown or killing himself over the flu vaccine.
Why is that?
Because it's even worse, right?
Yeah, you don't die. If you take your flu vaccine, you don't die, right?
But the percentage of people who get vaccinated and then who get flu is much larger than the percentage of people who get vaccinated and develop disease following infection with SARS-CoV-2. Because many years we say, the flu vaccine isn't very good this year. Remember that?
Yeah, sure.
So then this person must really be super disappointed and depressed, sticking their head like underneath the blankets and not coming out until spring because the flu vaccine is way worse.
Yeah, the point well taken is that people don't care about that because it's not a pandemic, right?
I guess, but it's still a disease, right?
I should call the book Right, Amy.
No, there is no Amy in the book.
The Enterophiles. Oh, but I said I have to dedicate it to you. I have to dedicate it to you.
You don't have to dedicate it to me. I think you should have a section called the Enterophiles. But
Vincent, you've been at Columbia for 40 years and they don't know who you are yet. What's wrong with these people? So I just got this incredible award a couple of months ago, whatever.
Like 8 weeks ago.
It's the Richard Ernst Award, and 3 people at Columbia congratulated me. None of them in my own department. I got an email from Rachel Gordon the other day, Amy, the doc who
That's
runs
lovely.
the micro-infectious disease course. She's always
So is
been
she
very
number
supportive.
3?
She's
So
number
she's
3.
number 3.
Yeah.
So there was Ian and Rod,
Ian Lipkin
and
and
I
Rod
don't count.
Rothstein.
I
Why
don't
don't you
count.
count? You could be number 4, Amy, but I
No,
told you about it.
because, well, yeah, but other awards people have contacted me for. Like the award this month, people contacted me for.
Anyway, I don't really care, frankly. I'm not complaining. I just find it weird that people at your own institution don't email you and say, congratulations on this amazing award that you got. So, that's why, in a way, I'm not so sad about closing my lab. But no more complaining. I love to have especially Dr. Griffin's update. Going to Botswana, a month of September. Triple vax. Do not want a 4th vaccine. Thoughts on not getting it? I think you could, if you're concerned and you don't want to be vaxxed, wear a mask on the plane. That's the crowded situation where you're most likely to be infected if you don't want to be infected. And many people do that and avoid getting infected. Is it true that monkeypox is not for monkeys? It's absolutely true. It's rodent. It's in some kind of rodent, maybe multiple rodents. But it was first found in a monkey, so they called it monkeypox. But that's not the monkey that they found it in was accidentally infected from a rodent. Yeah.
Didn't somebody write to you all about this history?
Yeah, we're going to read that. We got permission to read the email. Did I tell you about that, where he said don't read it?
Yeah, that's why I said, did this person write to you about the history? Because he said don't read it.
Yeah, so we asked
So
him.
I wouldn't break his confidence.
He sent the
I wouldn't break
modified
his
version.
confidence.
He sent the modified version. He said we could read.
Good.
Okay.
Good.
Mm-hmm. I recently had a friend and family fly cross country with full-on COVID. Oh, despite all the info, they shouldn't have flown with full-on COVID. That's not nice. What do you think of that, Amy?
Not nice.
I don't know if you're seeing these comments, Amy. You look so happy and pretty. All the best in your new career adventure. I just want you to see how people care for you.
It's good.
How are SARS-CoV-2 T cells detected in the lab? Well, you take lymphocytes you get blood from a person, you take out lymphocytes, you put them in culture, and then you add peptides, short pieces of protein derived from the virus, and if there are T cells that recognize those, the T cells will begin to proliferate and make molecules that you can detect, and that's how you do the assay in the lab. Clinically, I don't think clinically that's done. Is it, Amy? Clinical labs typically don't. There is one T cell test that can be done in a clinical lab, but it's quite rare because, as I understand, it's a little more laborious than a neutralization assay, right, Amy?
Yes.
Did I miss your coverage of whether we have established memory T cells? And multiple groups have found memory T cells, yes, have been established by vaccination, for sure. How long they last, we don't know. Same with the memory B cells. But they're absolutely being established. You can tell, you can look for the markers of memory T cells and people even, and you can find them. Yeah. Is there any issues for people with psoriasis? So, Chris, you emailed me about this today. It may exacerbate your psoriasis, but as far as Daniel has said, there's no reason not to be vaccinated because of that. What happened to Zika virus, Amy?
Its number of cases has dramatically dropped, but it's still circulating. Just nowhere near the number that we saw and stuff.
Yeah, it's still around.
Mm-hmm.
I get the pattern for the book. Virus Vinny. Yeah, that's it.
It's good. So you're going to start with virus because virus is older than you?
Oh, I don't want him to be so self-absorbed, okay? Let's put it that way.
I don't know that I would consider you. I wouldn't consider you self-absorbed, nor would I
I think
consider
if you're,
starting off with Vinny self-absorbed. I think it's fine.
I think you're right. A memoir is kind of self-absorbed, but I
I
think it
don't think
would
so. Who
look.
told you that?
You could
Who
imagine
told you
chapter
that?
one. You could imagine chapter one. No, I'm just saying it. Nobody told me, Amy. You can imagine chapter one opens on a quiet street in Patterson in early 1953. Blah blah blah. That's me. Or you could,
Really? You think that that's you? You're not sure.
or
Do you want
I
to
could
get
talk
back to
about a
me?
virus somewhere, right? On a quiet
Well, you
street
could,
in 1903, a virologist worked blah blah blah. It could work either way.
or you could start off with Albert Sabin and I lived in the same town.
Yeah, he did live in
Albert lived
Patterson.
in
Yeah,
Patterson.
yeah, I heard that. I heard that. And I mean, Albert Sabin's letters will factor into it because he and I corresponded. Yeah.
How could Albert not be factored into it? Albert is Albert.
Have you
Everybody
reactions
needs
to
an
this
Albert
new
story.
study saying repeat COVID correlates with bad outcomes? Yes, absolutely. I saw that. Amy sent it to me this week. Right. So repeat COVID, higher likelihood to have long COVID. Not actually surprising, right?
No.
I think it makes sense. Amy's move has done her so much good. Yes, absolutely. In many ways, I think she will live longer as a consequence. Let's put it that way.
I'll live longer.
Yes.
I didn't know I was dying faster. I didn't realize I was dying faster. Okay.
Gee, we're just getting to the Amy is always right comments. Oh my gosh. Which Amy could write her own pandemic book. She could, but she doesn't want to. She wants to spend her time doing experiments, right?
Yeah.
There have been a lot of public health, a lot of improvements in technology that coincided with COVID. I think we'll see a lot of improvements in tracking and coordination, hopefully, yeah. Well, I agree.
Where's the money going to come from? Where's the monetary investment going to come from?
Well, that's a big problem. Public health needs money and governments don't want to invest it. Real problem. Vera. Ooh, a good name. Vera or Vira. Thank you for your contribution. I would say Vira, but you may say Vera. Yeah. The world according to Amy. All right, so now we have some questions for Paul. All right, I'm going to have to go back to the stream and copy these down. Why I shouldn't get a second booster. With icosahedral viral structure, does a subunit always have 3 proteins?
No.
It could have 1. The simplest particle is 1 protein per subunit. Yeah. Here's another off-it question. I won't go through all of these, but I'm going to go back later to the stream. Ask him about the butterfly effect. Are you serious? That's when you kill a butterfly back in prehistoric days. It changes everything nowadays? I don't think so. I don't think that would be good. Reading now that BA5 is more severe, that it goes deeper in the lungs. No, don't believe it. It's not a reason to be more concerned about contracting it. I don't think as we have already said, I don't think any of those studies are valid. Virologists are assay pushers. We are, aren't we, Amy?
Yes.
What's your favorite assay?
Plaque assay.
Not mass spec? No. I have to claim influencing you with the plaque assay, okay? You can't deny that, young lady, okay?
I would never deny that. My lab is now changing over and they're traumatized.
Do kids 6+ need the 3rd dose? That's a good question. Yep. That's a good question. In the Andromeda strain, a wildfire team of experts was assembled to secretly identify and mitigate the infectious agent to save the world. Would you guys volunteer? Sure,
For
I
sure.
would, but I'm too old. They don't want me. They just want me for consultation. Did I ever tell you, Amy, that I auditioned for one of these doomsday TV shows? What the hell was the name of
What do
it?
you mean a Doomsday TV show?
A post-apocalyptic TV show with, most of humanity is killed and you have to survive or something.
Okay.
Yeah.
Never heard of this.
The Colony. I auditioned for The Colony.
What is the colony? I never heard of it.
I don't know. It's a TV show.
Okay.
And
Wow. I'll have to look it up because I don't know what you're talking about.
yeah, it's a post-apocalyptic thing. And these people are in New Orleans and they have to survive. There's like 7 people or something. And it's weird. I said I can't take time off to make do a 25-episode TV show. I just can't do that.
Crazy.
Also a question for Offit. Any studies looking at increase in inflammatory syndrome in people post-SARS-CoV-9 vaccination, especially patients with autoimmune disease? I haven't seen that, but I can ask him that. A long time after vaccination, memory cells are expected to be a main player. What changes in the cell should explain the slight decrease in protection against severe disease after a longer time? Well, I think memory cells don't last forever, right? And that could explain part of it. What do you think, Amy?
Right.
Yeah.
I think that's true.
Oh, my laptop is about to run out of juice. Hold on.
That's not a good plan.
I have to plug it in. I thought I had plugged it in. Oh, it's going to run out, and I just don't have a cable here. Sorry.
That's not a good plan.
Well, I came from Princeton, and I got kind of wrapped up in the other recordings, and I forgot to plug it in.
And I can't believe on the power strip of power strips you don't have a power block. It's hard to believe.
I do, but the cable's too short to reach. And I'm trying to think here what I can do.
Maybe you should move the computer towards the cable.
Well, then I can't see it. Thank you, Pamela, for your contribution. Is it true we have to be cautious even outside with BA5? I don't think so. I think if you're triple vaccinated or quadruple, you're fine. You're reading John Barry's The Great Influenza. Yeah, I got it right here on my shelf. John agreed to come on TWIV, actually.
So? You should follow up.
I never followed up. There are a couple of people I have to follow up with. And he's one of them. Will there be a cure for Hep B like there is for Hep C? Why don't you answer that where I try and plug in my computer, Amy?
I don't think so, because Hep B is very complicated. Plus, the polymerase is totally different. It has to do RNA and DNA. So I don't think so. But I could be wrong.
Well, also the problem with Hep B is that you have that double-stranded DNA which becomes episomal in the cell, and you can't get rid of it. Antivirals don't get rid of it. Whereas Hep C, there's no such stage. So
but there's like a pseudo cure for like herpes, even though the DNA is episomal, and that's acyclovir, right?
Yeah, but there's not a cure. The acyclovir does not get rid of the latent herpes DNA in the neurons. It just gets the cold sore to go down, right? So it's the same problem.
I know.
So I plugged it in. Now the laptop is 4 feet away from me, and my old eyes have trouble seeing.
You'll be fine. Your old eyes.
The Colony. Les says the Colony had the guy from Lost who played Sawyer. Yeah, they picked someone else.
Who?
They should have picked me. Did you see Did you see Lost? Did you see Lost?
Lost? That's a TV show?
This is from the lady who never watched Star Wars.
Yeah, exactly. I don't understand why this is even a conversation. I mean, you know the answer is going to be what? This is a TV show?
What's the likelihood of catching COVID from swimming indoors and sauna use?
Small.
Small. The sauna is really hot and the swimming has chlorine.
Exactly.
Are people getting infected with SARS-CoV-2 more frequently than with flu, even though they're vaccinated? So that's a good question. I think we're testing a lot, so way more for SARS-CoV-2 than we do for flu. So I don't think we have a good estimation of flu reinfections. What do you say to that, Amy?
I agree with that.
Yeah, so I don't know the answer, and if we did more testing for flu, we might know. Okay, here's another question for Paul. Do you think people with COVID are infectious after 10 days? Good question. We'll ask him that. And Toga virus, the virus that spread through Delta House, and John Belushi had it first. That's very good. I don't know if you saw that movie, Animal House with John Belushi. They have a toga party, and they're all chanting, toga, toga. Did you see it?
No, I didn't see it. No.
Did you see any movies, Dr. Rosenfeld? Name one movie.
I've seen Charade.
I knew you were going to say Charade. I knew it. Did you see Breakfast at Tiffany's also?
I did. I saw Catch a Thief.
I
saw Notorious.
Did you see any Alfred Hitchcock?
Catch a Thief and Notorious are Alfred Hitchcock.
What about adeno-associated virus? That was on my list. Yeah, for sure. We're going to do that. Vanity, occasional mention of pets. Yeah, I would mention pets, even though it would be a human-centric. We're going to have pets because they interface with humans, right?
Yes.
What does a virus try to do? It doesn't try to do anything. It doesn't try to do anything. It has no emotions, no directed thing, nothing. It's all passive chemical reactions. The only things viruses do, if you have to say do, is reproduce. That is their modus operandus, reproduction. Say toga, toga, toga. See, that Mark from Australia knows about Animal House.
Look, I never said I was a movie buff.
I'm not giving you shit. Don't worry. I'm just kidding.
It's fine. I don't mind. It's fine. I don't mind.
When will we have a sterilizing vaccine? Yeah, I think that's a common...
There's no such thing as a sterilizing vaccine.
Charade is the best Hitchcock movie not made by Hitchcock. That's great.
That's true.
I didn't know that.
True.
Yes, there will never be a sterilizing
vaccine.
I mean, Daniel brought this up today. He said maybe we should be working on sterilizing. And then you have to change the immune system.
What did Daniel say?
He said we need to work on vaccines that prevent infection. So he didn't use sterilizing, but he did say prevent infection, which is not happening.
That's the most ridiculous thing I've ever heard him say. He needs to go back to medical school. That's the most ridiculous thing I've ever heard.
For viruses live, do people need to take or review? I would either take or review it, but you took it, Peter. I remember you were there, so you should be good. Is it true that a single Moderna jab offers sufficient protection to have passed the FDA minimum target for 50%? Yeah, it's true.
Mm-hmm.
They would have licensed it at 50%. In fact, Peter Marks said that a long time ago, Amy.
I know.
Here's one for Amy. I'm going to let you answer this. Are you ready? What do you think of Bob Wachter's line of thinking? COVID, BA5 easily causes more reinfections and potentially HIV-like damage to the immune system, T cells. Question. What do you think of that, Amy?
I think he needs to step down his chair of medicine and give back his MD. Or enroll in an infectious disease course.
He's an internist. He's a medical doctor.
Well, that's what I said. He needs to enroll in an infectious disease course, or he needs to step down his chair of medicine and give his MD degree back, because that's just wrong.
Did you see North by Northwest or The Third Man? People want to know.
I saw North by Northwest, but not The Third Man.
Zika's in the course. It's a flavivirus. Yeah. Yep. Yes, it should be human-centric because otherwise it's too much. So what do you think of this? I could do another course next year on viruses of plants and bacteria and fungi. That would be cool. And another course on viruses of non-human animals. I could teach so many courses, folks. Are you in it with me? Are you in it for the duration? That's the question. Because you 547 of you are my audience.
I'm here.
I'll do what you want. I know you are. Is COVID a super virus that saps humans of their disease-fighting capabilities with every reinfection? No, it's not. Immunity works quite well in most people at taking care of SARS-CoV-2. So I don't know if you've got that impression from the press. I'm sorry, because that's not the case at all. Lisa says for Offit, what's his greatest concern with next-gen vaccine development? Okay, that's a good one. Please ask Paul what Brianne was saying about risks. Oh, what risks are there if you get boosters? Yeah, well, let's ask him what's the risk of having a booster every 6 months. How's that? I like that.
I think he once talked about the potential for autoimmunity.
What about, where are you wrong about boosters in over 50? Are there T-cell tests to test value? Okay. Jeff wants a course on just plant viruses. Yeah, because you work on them. But as I said, we could have a separate course. Question for Paul. Do we know the difference in T-cell memory comparing 2 doses versus 3? Okay, that's good. That's good. A lot of Paul questions. Thank you so much for them. I'll take note of them. Vincent, eradication refers to disease no longer being around, not a virus. It's right. Is it right to say that even though SARS-CoV-2 Delta or flu B Yamagata are no longer around, they have not been eradicated? Well, you know,
so
the
first
WHO...
of all, wait a minute. First of all, eradication in terms of polio is the virus, not just the disease. It's both.
Yeah, you know, in the old days, before you were born, it was originally to eradicate the disease. Then the WHO expanded it to include the virus.
Yeah, I know. I spend a lot of time with Kostya, and he's not happy about that. They should have just focused on eradicating the disease, and then some brilliance came up with the virus, which is a total disaster.
I don't know what Hans is saying. They have not been eradicated. Well, the virus is gone. But the problem is that saying a virus is gone is only as good as your surveillance for the virus. So you really never know, right? You could say polio is eradicated, right, but then it could pop up in 5 years. Next case.
Yeah.
Go with Novavax over mRNA vaccines? I don't know why. The mRNA vaccines are well proven. They work really well. If you're afraid of myocarditis, is there any myocarditis associated with Novavax, Amy, that you know of?
Not that I know of.
But, you know, Nova's not been in as many people as the mRNA vaccines. I read that BA5 has an R0 of 18. No idea if this is true. It's wrong. No way it has an R. We were talking about this at the meeting today. Amy, you would be impressed. They all agree that R0 is nonsense and also herd immunity.
Wait a minute.
They're channeling Amy.
Wait a minute.
Go ahead, talk.
I am stupefied.
So Theodora said, oh, the latest is that BA5 has an R0 of 13, and she laughed. And this is just nonsense because they don't know how to calculate.
There is no such thing as an R0. It's not an intrinsic property of the virus.
Okay.
Oh, my God. I might even read this book.
Some good suggestions for the course. You could be selective, and I'm going to have to be somewhat selective. Yeah, principles. I like that. Contributed some important understanding of a principle. That's good. Do it over 2 semesters of 22 weeks each. So the problem with that is, this is theoretically a course beginning in September. It needs to end in January because then I teach my Columbia course. Could I teach 2 courses in a semester, Amy, like concurrently?
No.
Why?
Too much work.
Too much for
Too
me?
much work.
You're saying
Too
I'm
much
too
work.
old to be able to do it? Huh?
No.
You're saying I'm too old?
No. Where did you get too old? So let me get this straight. You want to teach 2 courses in 1 semester and do 6 podcasts. Good luck to you. Let me know how that works out to you. Let me know if you're still married at the end of it.
This is my life in the incubator, Amy. That's the whole point of closing the lab and doing full-time teaching in SciComm. No?
Okay. Let me know if you're going to still be married at the end of it. Because I'm thinking no.
Okay. All right. So, no herd immunity. Vaccines help from hospitalization, but long COVID is a possibility. All right. So, I think Gout is listening to Lori Garrett, right?
Sounds like it.
The vaccines substantially prevent deaths. They reduce severe COVID. And they reduce long COVID to maybe below 5%. Those are the numbers that I'm looking at. So listen, Gout, many people choose to mask because of these reasons, and I fully respect that choice. But I look at it differently. So I'm not masking. I read that, oh boy, a non-COVID question. Amy said before this started, I hope we have some non-COVID questions tonight. I read that mosquito-transmitted flaviviruses can manipulate host skin microbiota to produce a scent that attracts mosquitoes by suppressing expression of a skin antimicrobial protein. I haven't seen this paper.
True.
Is this a new paper?
I think it's a couple of months old. It might have been lost, but I believe that is true.
So they manipulate the microbiota. But why would they?
Well, they reduce the infected cell does exactly like what Alice Prince thinks some infected cells do. They change the metabolite, and then and this is what GERN does too for rhino. They change some kind of metabolite that then changes the way that the bacteria interacts with the cell and with itself, like glutaraldehyde or glutamate or something or other. Yeah. Alpha-ketoglutarate is Alice Prince's molecule. Yeah.
That's cool. It's very cool. I'd like to see this paper. We might do it, Barb Mack.
Yeah, it's very cool. I'm sure I sent it to you last month.
Split the course. I'm thinking about it. Amy thinks I can't do multiple pods and courses at the same time. But I'm thinking I have no lab to go to anymore, so...
Okay, so do it. Go ahead. Do it.
I don't think there's any
just that
way...
It's just going to take a long time to generate the first version of this new class.
I understand. Well, that's why maybe spreading it over 2 semesters would be a good idea.
Maybe.
I don't think there's any feasible way to address an R0 for BA5. We are not a naive population. Well, the epidemiologists think.
There's no possible way to describe an R0 even for a naive population. It's a made-up number. It means nothing. As I said, my EV71 has an R0 of 36 in a daycare center.
Well, no,
kids like to snot each other.
So let me get this. Let me make sure I'm understanding this. So you are saying all the epidemiologists in the world are wrong by calculating R0s in naive populations. Is that correct?
Yes. Saying that there's no such thing as an R0.
That's really a strong accusation. I wouldn't be so strong, but...
Really? Considering the fact that you guys all think that picornas don't spread, picornas are not very transmissible. You would say polio is not really very transmissible compared to measles. And yet, in a daycare center, the R naught of EV71 is like 35, because every 3-year-old likes to like take their mucus and smear it all over each other.
Here's Alice again from Ireland. I love how she writes. Both looking good. Where do you get the energy? From you guys.
It's good.
Hey, we got 300 likes, 540 people. Could you just hit the like button to get that up to, say, 400? We'd appreciate it.
Good.
What was this question? Can people that clean hotel rooms get monkeypox from the bedding and from towels?
I don't think it's transmitted by fomites.
No, it can be, but they would have to rub it on them, right? So they're wearing gloves. Do people who clean rooms wear gloves? And they're holding it at a distance?
I don't know, but they wear some kind of clothing.
I don't think it's an issue. Now, smallpox you could transmit by blankets, but I think...
Well, we did that.
That's partly...
But nobody was wearing the clothing.
Also, they're probably inhaling the fomites, the aerosols generated, the dust generated from the blankets, right?
Yeah, but that's the famous thing that we did in the French and Indian War, was we gave the Indians all smallpox contaminated blankets. We knew that that's what
right.
we were doing. It is a disaster of the Caucasian man. I mean, no wonder why people hate us. Unbelievable.
How likely is it that I could take 2 rapid antigen tests and 1 PCR test, all negative, and still have had COVID? How do you know you had COVID if all the tests were negative? So Jane Flint has the remnants of influenza right now. And she, I said, are you sure it's not COVID? She said, no, I tested negative multiple times. So you could have something else. As Daniel always says, test prudently and remember there's more than COVID out there. Is it test prudently? I'm not sure if that's the word he uses.
Well, now he uses it, but it wasn't before.
Look at that look from Dr. Rosenfeld. The arched eyebrows, you know? Here's another Offit question. Are we not developing T-cell capability at all? I think we are, frankly, but I do want to ask him about T-cells. Did you read anything on COVID attacking T-cells? Yes, we have, and we think it's it's a cell culture observation. Not sure that it applies to most infected people. What's an alveolar cell, Amy?
It's the cell that gas exchange occurs at in the lung.
It's way down in the depths of the lungs,
it's at
right?
the very bottom. Yeah, it's the little balloony cells.
Are you going to discuss the rising death rates compared to previous 5 years? Well, there are all-cause rises in death rates. We have discussed that on TWIV, actually. I don't know what else you want to say, but besides COVID deaths, then other causes are increasing as well, because COVID can exacerbate other conditions, for example. Environmental loves your camera and lighting, Amy.
That's good. This is all courtesy of Richard.
Richard
Lund.
From California.
California.
She provided the lighting for Dr. Rosenfeld. Yeah.
Yes.
And you have a couple more from him that you're going to deploy subsequently, right?
Yeah, I do. I haven't been home to assemble them, because I picked them up in New York, right, on Wednesday, and then I went back to New York on Friday, and then I came home on Tuesday, right?
Mm-hmm.
And today is Wednesday.
Yeah, Amy's been driving around a lot in the last couple of weeks. Anyway, Marco suggests you visit
and now I don't want to drive anymore.
The Bata Shoe Museum online. Maybe you want to check that out, Amy. Okay.
All right, I'm going to check it out. Gotta go to see the shoes for sure.
How about Florida not ordering vaccines for under 5? This is absurd. Florida is wrong on this. Very wrong.
Yeah. And then he let them order. Then he didn't interfere. Then they ordered.
Paul offered at Brown talked about the incubation period of SARS being too short for any vaccine to prevent infection. Memory takes too long. Yeah, it does. It takes 2, 3, 4 days, right? And exactly why you're going to get symptoms. But then once that memory kicks in, your symptoms subside in most people anyway. In Easton, we have a shovel museum. Amy, do you want to go to the shovel museum?
Not particularly, but if you want to go, I'll go.
No, I'm just joking. I didn't think you.
I went to see a whole
want
series
to
of
do that.
I went to a whole exhibition that filled the Cooper Hewitt on thermostats.
Thermostats would be cool.
The guy thought it was super cool. He convinced us all to go.
Maybe smallpox vaccine was keeping monkeypox under control. Yeah, I mean, we haven't vaccinated most people against smallpox for a long time. I mean, the military, for example, still gets vaccinated, and you could argue it just is wearing off maybe, but I think it wore off 40 years ago, frankly. So Daniel said today there was an outbreak 20 years ago of monkeypox in the Midwest of the U.S., and many of the patients were vaccinated. So even then, it had already waned. What's going on now is unusual. And as we said before, not sure why it's happening. Amy, how do you get your teeth so beautifully white? Maybe it's natural. It's natural?
Yeah, I don't use whiteners. They're not good for your teeth.
Could polio vaccine be keeping enterovirus infection in check? Amy's going to love this question.
I'll get back to you in 5 years when I finish the experiments.
That's exactly what Amy's working on at the Food and Drug Administration.
That's one of the stuff I'm working on.
It's going to take 5 years. Aren't you going to know next week?
I'll know tomorrow, but I'm keeping it secret for 5 years. Where's the suspense music movie?
It's a good question, Kat.
When you need it.
It's a good question. Really good question. This is, and so if you stop vaccinating one day against polio, maybe you're going to get even more enterovirus infections, right?
Yeah.
Anyone recover from long COVID?
Now that we've changed.
Yes. People do recover from long COVID. Right now the definition is symptoms beyond 8 weeks after symptom onset. And there are people who recover where things get better. Their sense of smell and taste return, whatever other issues they have. Not everyone recovers, but some people do. And so it will be very interesting to know as we move years out from infections, what fraction resolve. It's a really good question. And so, Amy, you said the naive population is prone to monkeypox. How effective is my dose of vaccine from 50 plus years ago? I just don't think it's effective at all any longer.
I thought that Condit thought it was like lifelong immunity.
I don't think so. Because of this outbreak in the U.S. Go ahead.
So would you say that polio immunity is the longest then from vaccination? Because you think it's like 40 years at least.
I think it's at least 40 years, but it's been 80 years now since, when did we stop? 1980, so that's roughly 40 years since we stopped vaccinating. Yet
I didn't get vaccinated against smallpox.
No. So I was vaccinated in the 60s, I guess. So
I would say in 1971.
I don't think my vaccine is protective any longer. No.
Maybe we should ask Condit. He's the pox expert.
Do you have any insights into why the test positivity rate is considered significant. I don't know. You mean to the number of positives in an outbreak to understand when the outbreak is subsiding? Is it? Do you think is that what he means, Amy?
I don't know what he means. I have problems with this anyway because the testing rate varies, so it's not consistent. So, like now, you're probably the only people who are testing who are people who think that they have symptoms, right?
Yeah.
Whereas before, you needed to test
Well,
everyone, right? So you had one. So you had to. So here, it could be. It's going to be skewed, right?
but how else can you understand how
Well, if you
much?
randomly test, right? If you took one policy and said, we're going to test 100,000 people who pass through this turnstile
Mm-hmm.
at the beginning of the pandemic, and we're going to test 100,000 people that pass through the turnstile, then you have a random population, right?
Right.
Whereas if you just base it off if you just do it the way we're doing it now, where people only test because they think that they have symptoms, you have a skewing of the you have a skewing.
But you've always argued that you don't want to be testing randomly because that gives a false
Well, I don't
impression.
want right, but I've argued that you don't want to test randomly. You don't want to continuously test because it's not helpful in making social policy. But if you want to know how much is actually circulating, that's a separate question, right?
Yeah. Yeah.
So I argue that social policy should not be based on testing. But if you want to go out and figure out how many people out of your 100,000, how many people have SARS-CoV-2 infections, how many people have flu infections, how many people have both infections, then you have to test randomly.
If a person can't hold immunity to rubella, would that correspond with mRNA vaccine? I don't quite understand that. The rubella vaccine is an attenuated virus vaccine. It's not an mRNA vaccine. And so, but
I
there's no
don't know.
relationship, right? It's a different vaccine, so you really can't compare the two in terms of longevity, if that's what you're asking.
Yeah, I don't know.
Long COVID damages blood vessels. What does that mean? In people with long COVID, one of the things they have are damage to blood vessels, possibly caused by clotting or inflammation. Inflammation does things to blood vessels, and it's persisting. Why it is, well, actually, we don't know the cause. I'm just speculating. But why it persists, we don't understand either.
Well, I don't think blood vessels. Excuse me.
Okay, you got to repeat it.
I don't think blood vessels regenerate. It depends on what kind of blood vessel you're talking about. First of all, I don't believe that like arteries and veins regenerate. Maybe the cells in the capillaries do, but arteries and veins, I don't believe regenerate.
Well, if you have a tumor, it gets vascularized, right? New
That's
vessels
new.
grow.
It's new vessels that are growing as extensions from old vessels. But there are capillaries and venule and arterioles or venules. I'm not even sure that they make it all the way to arterial and venules.
But
I
if you
think that
cut,
they're just
you cut yourself, you cut capillaries, they heal and they reform their
yeah,
anastomoses, right?
yeah. But I don't know that the damage that you're talking about when you talk about blood vessels is to capillaries versus larger vessels.
Yeah. Do you have a new mouse house, Amy?
I do.
Is it nice? Have you been?
I've been once, yes. It's very nice. I passed training 1 and 2 yesterday, and I have to write to figure out what to do for training 3 and 4 because it's some kind of complex thing and you need a registrar and it's I don't know. It's too complex for me to figure out. I don't know how to register because you're supposed to have your organization on the front page, but the organization isn't on the front page. I don't know. It's very complex just to get past the
I'm
front
sure you'll
page.
be okay. I'm sure you'll be okay. Do
I'm
you know
sure.
whether cannabis is susceptible to viruses? I'm sure it is. Everything has a virus or
I
multiple
believe it
viruses.
is, and then it's susceptible to mold.
Now, wait a minute, young lady. How would you know this?
My friend is an accountant, and he has clients who are cannabis growers in Canada, and it's very specific.
I
And
see.
I also, if it's too wet, you get a lot of moldy pot plants. We
Not
can
good.
ask other people.
Is anyone doing surveillance sampling in monkeys like the U.S. is doing in deer?
Why would we be surveilling monkeys for what?
I think SARS-CoV-2 because you're talking about deer where we're surveilling for SARS-CoV-2, right?
Oh,
I'm not
okay.
aware of it. No,
No, I'm not
not at all.
aware.
I'm guessing this guy doesn't care about long COVID. Now, why would you say that? I just answered your long COVID question. We just answered your blood vessel question, Stanley, and I answered someone else's about long COVID. So that's
I
not
think it's
fair.
just mean and rude.
It's not fair. Affects everyone but does not infect everyone. That's right. That's what we said. Amy doesn't like it, though. She doesn't like it.
Then everything's a pandemic.
Well, in some cases, we have a pandemic of rudeness, right?
Yes.
Paul Offit on NPR today said a pandemic is measured by the amount it disrupts normal life on a global scale. That's an interesting idea, isn't it?
Yes.
Uh-oh. Are you ready for this one? WHO
I'm
announced
sitting
today that
down.
monkeypox virus has mutated. It's so
Really?
stupid. It's so stupid, Christopher, and you know it because viruses are always mutating. Now, do they have proof that a mutation is responsible for the altered pattern of infection? I doubt it. I very much doubt it.
Oh, you wanted experiments? I don't
Yeah,
think
I
we do
do.
experiments. I
Do you
think we
think
just talk to the virus.
do you think everyone will get COVID?
I think everyone will be infected by the virus. Now, whether or not they develop symptoms and disease that is COVID is a different question.
Good, I agree with that.
That's my answer.
Yeah.
And I'm sticking to it.
Thank you, Westfield, for your contribution. Do you think preprints are a good thing or a bad thing? They're good for scientists. We get to see things ahead of time. And I always thought they were good for everyone, but as you see, the press gets them, and they're not quite sure what to make of them, and they get into headlines, and I think it's misleading, and it's a problem. I just agree with that. Amy, is the common cold a pandemic?
Yes, by your definition. Everyone's affected, but,
Yeah,
not everyone is infected. It's a pandemic.
But it doesn't disrupt normal life on a global scale, rhinoviruses.
Well, it does. It does.
I don't think so. I don't think so.
The economic burden of rhinoviruses is a rhinovirus infection outweighs the economic burden of almost everything else.
Really?
Yeah. It's almost, it's about $100 billion a year. Yeah.
Well, maybe you should tell the WHO about it because maybe their internet is broken.
I don't want to talk to the WHO.
And
I don't want to fix their internet. They can fix it themselves. They have an IT department. I could barely fix my own computer at the FDA. And I had to have like 400 people come and fix my computer. It took 12 days.
One of the large national labs have now ramped up for testing for monkeypox. Yeah, Daniel mentioned that. Now you can order a test, and that's great. All right, Patricia's
That's
coming
good.
back from repairing a bathroom window to answer the HIV. Casual sex. Everyone has to worry about HIV who's contemplating sex and needle sticks. So you asked, how does that affect your life? So Patricia's answering that, okay?
Yeah,
What do you think?
but children don't get children aren't who are not sexually active before puberty are not worried about casual sex. Most married couples are not really worried about casual sex, right?
Yes.
Okay.
In a
And
pandemic,
really, do you really think that the senior citizens who are not using condoms are worried about HIV infection? Or do you think that they're still having random sex and not using condoms? And that's why syphilis is running rampant through nursing homes. Hmm? Hmm? Hmm?
yeah, well, this is a problem. Sex is surreptitious. People never admit it or rarely admit it.
Okay.
In a
So,
pandemic, is the pathogen always new to us?
generally.
I mean, it's new. We discover it, but it may not be new. It may have been around before, right? But yeah, I think that because if you look,
But I
HIV,
think
COVID,
generally
Hep C, Hep B, influenza pandemics, at some point we discover it.
well, generally, yes, but generally, it's new to us in terms of immunology, right? We're I would say it's new to us because it's an immunologically new agent, right?
Yes, it is. It's a new virus
So I
also.
think actually that's a better definition for a pandemic
What's that?
is that it affects a significant percentage of an immunologically naive population.
What's a significant percentage?
50% or more.
Where do you get that number from?
Because everything, a majority is always considered 50% plus one.
Okay.
That's what a majority is considered.
So Salvador says, so you are saying senior citizens are having sex?
Yes.
Shocking. Shocking.
Who would have known? Oh my God. I
Hans
can't
says,
believe.
Hans says, Amy, after the multi-organ damage that SARS-CoV-2 is clearly able to ensue, is it likely in your view that many, if not most, human diseases have a viral origin at some point?
What?
All human
I
diseases have a
guess.
viral origin. That's the question. What do you think? Yes or no?
I don't know that all human diseases have a viral origin. I
No,
would say
I don't think
50%
so.
plus one have a viral origin.
Now you're going to get that. All right, listen, I promise when we make the pandemic definition, we will consult you, okay?
You don't need to consult me. I'm not technically an author of the book. I just think you guys need to sit down and really think it through and say, you know, do other things fit this category? And other things fit this category, but they don't get a lot of press. It's 8:58, and I'm getting cold because the air conditioning is turning on. And
Yeah,
they want to.
9:58. We're going to wrap it up in a few minutes. No worries. Recently, you said you received the Tdap. I'm 69, no health issues. Is this recommended? A Tdap, I think, is every 10 years, right?
Yes.
Yeah, you should get it if you go to your physician and they keep records, they should say, oh, you're due for a Tdap. That's what they do with me.
Yes.
Okay, so let's let me do this. They can't bud out of the plasma membrane because of the cell wall. I'm not sure about that, Jeff, because you can make influenza virus-like particles in plants by simply producing the HA protein, and you get virus particles budding from the plasma membrane, and they stay below the cell wall, right? They stay restricted. That because you get those particles in plants with influenza virus. All right, there are a lot of good questions here, but Amy is tired and cold, so we're going to wrap this up. I'm not going to throw Amy out, though. She just left the lab. That was a a reference to that. Thank you, Westfield, for your contribution. Recently, I'm hearing too many saying we never expected that regarding virus mutating, blah, blah, blah. Well, yeah, we didn't, as Amy said, because we never looked in the coronas for antigenic variation. And when we did, we found it. So I think they should have expected it. Yeah. Siddhartha, thank you for your contribution.
I was trained that everything works the same and you plug it in until you find an area where it doesn't work the same.
Is it true that WHO never declared COVID a pandemic? That's not true.
Yes, they declared it a pandemic.
Condemn it a pandemic. It's in that the declaration. The guy says this is a pandemic. I'm pretty sure about that. All right, let's go through the contributions and we'll wrap this up here. A lot of good questions. Boy, I'm always sad at wrapping this up because we get so many questions. Thank you, Curly Dog, for your contribution to the incubator and to science communication. And next time I won't talk so much in the beginning.
You can talk as much as you want.
So we can get more questions. It's no problem.
You can talk as much as you want. It's good.
What episode was the man on recently talk about 1918? Well, he wasn't on. He canceled, so I have to revisit it. I'm hoping to go interview him in August. Right? Mm-hmm. Oh, so many questions here. Holy cow. Elena, thank you so much for your contribution. Really appreciate it. And lots of questions for Paul Offit. That was the biggest comedy moment when you and Amy, when you were touching your tonsils. I got it. Just lymph nodes. I know, but I always, when I get a scratchy throat, I palpate them and I say, yeah, they're bigger. So I know the size of my lymph nodes down here. Do you know the size of yours, Dr. Rosenfeld?
Yes, and I have many people monitoring them.
Elena teaches 2 courses every semester, but I do no podcasts. I do a lot of podcasts. Yeah, but I could do it. I can do it. That's the whole point of the incubator, is to do it.
It's good.
Thank you, Old School, for your contribution. Really appreciate it. To science communication in the incubator. If any of you are around New York City, come visit the incubator.
Visit.
Come visit. All right, folks, that'll do it for another Q&A. Thank you to our moderators. Les, Steph, Vanity Nutrition, Tom, and Frank were here tonight. Thank you all for coming. 540 of you. By the way, we passed 111,000 subscribers on the YouTube channel. How about that? Isn't that cool? Moving forward.
Great.
Moving forward.
Moving up. Moving up.
Bring back your questions. Now, wait a minute. Next week, there may be an issue next week. No, that's the week after. Next week we'll be here. Oh, no, I'm flying to... what is next week? What's the date? Today's the 6th, and the next is the 13th. Yeah, the 13th is good. The next week, I am flying somewhere. I'm flying back from Wisconsin. I don't know if I'll make it in time for Q&A. We'll see what time that flight gets in. Anyway, folks, have a safe week. See you next time. And thank you for coming this evening. Good night, everybody.
Bye.